Dear students, in this lecture we will study carbohydrases. This group of enzymes hydrolyzes polysaccharides or oligosaccharides. You know what are polysaccharides. These are long chain of glucose molecules linked together, linked through the glycosidic bonds. Uh, they can be branched or they can be straight chain. So we have this group of enzymes and it includes the alpha amylase, beta amylase, which I have not mentioned here, beta amylase. We have glucoamylase, we have invertase, lactase, glucose isomerase. So let us discuss them one by one. First of all, let us discuss alpha amylase. This alpha amylase is capable to break down the 1,4 linkage, but it is not capable to break down the 1,6 linkage. So as you can see in this picture, this is the amylose chain which is straight, and these glucose molecules are linked with each other through glycosidic bond, which is 1,4 linkage, and the second chain is branched, which is amylopectin, which is having the 1,4 linkage as well as 1,6 linkage. So this alpha amylase will be capable to break down the 1,4 linkage only, but it will not be able to cut this bond. It will not be able to cut this bond. So this alpha amylase can act at any point. This is very important point. You should remember this. It can attack this chain at any point. At any point, and it can break down this chain at any point except the 1,6 linkage. And the end point, end result will be the maltose, which is a disaccharide. So if this alpha amylase is going to act here, it will cut this dimer. And now there is a question why alpha amylase cuts the dimers, why it is capable to cut dimers only. It is because of its configuration. Its configuration does not allow it to cut single units. So because of its configuration, it cuts the chain into dimers. So the end product will be maltose, and it will liquefy the starch. That is why it is also known as liquefying enzyme. Let us discuss the beta amylase. This enzyme is exoenzyme, and it also produces maltose units just like the alpha amylase. But here one thing is different, that it is not capable to attack at any point of the chain. It is not capable to attack the chain at any point. It can cut the chain only from non-reducing side. Suppose this is the amylose chain. Here it is the reducing side, and here it is the non-reducing side. So this beta amylase can act only from this side. And again, this enzyme is not able to cut 1,6 linkage. So in case of the amylopectin, it is not able to cut this bond. So here we will get the two end products, maltose and isomaltose, because there will be 1,6 linkage. The formula is same, but the linkage is different. So we will get two types of products in case of the amylopectin. One is the maltose, another is the isomaltose. Now the third type of amylase is the glucoamylase. It is also known as saccharifying enzyme because it increases the sweetness. What uh, this enzyme do, it cuts the chain from non-reducing end just like the beta amylase, but It can cut also the alpha 6 linkage. However, this reaction will be slower than the 1,4 linkage cut, but it is capable to break down this 1,6 linkage. 1,6 linkage. It is able to cut. And the end product will be the glucose, single unit. Its configuration allows it to cut the chain into single units rather than the dimers. So, uh, so the end product will be glucose, which is sweeter. That is why it is known as saccharifying enzyme. So here we have to remember that this enzyme is capable to attack the chain from non-reducing end, and it can cut the chain at 1,4 and also at 1,6, and the end product is the glucose. Now the another enzyme is the invertase, which hydrolyzes the sucrose, which is a dimer, into fructose and glucose. Uh, This reaction is carried out when we have to prevent the crystallization. Sucrose, in case of sucrose, it crystallizes easily. The products which are made up of the sucrose, they have the problem of the crystallization. So what we do, we convert this part of the sucrose into the fructose and glucose by using the invertase enzyme. By doing this, the solubility increases and the crystallization is prevented. In addition to this, the sweetness is also increased. Then we have the lactase enzyme, which is actually present in human intestines, and it breaks down the lactose, which is a milk sugar. This sugar is present in milk, and this lactase breaks down the lactose into glucose and galactose, these two monomers, and then these are utilized by our body. But in some persons, they lack this enzyme, and they are not able to digest the lactose. Hence they have a problem which is known as the lactose intolerance. So to overcome, to produce products for such type of persons, we use lactase. uh, we add lactase to the milk, and we convert lactose into glucose and galactose before the consumption, and by this way we produce the lactose free milk. Then we have the glucose isomerase. This glucose isomerase is used for the production of high fructose corn syrup, sweetener from glucose. Glucose is converted into fructose. This was all about the carbohydrates. Hope you understood this. Stay safe. Thank you very much.